share this chart. Thanks for coming, everyone. Today I'm going to talk about ASTRID, which is a fast method that we've developed for species tree estimation under the coalescence model. So what does that mean? What we're trying to do is we're trying to recover species trees from large data sets that have evolved with some sort of gene tree discordance. So a lot of times this means from whole genomes. So we start with our online sequences, then we use a multiple sequence aligner like Taxa, Muscle, or Cluster to get alignments. Then we use typically a maximum likelihood estimator to get gene trees from those alignments, and then we combine these into a species tree. And what we're going to focus on is this process of getting the species tree. So there are a number of desirable characteristics in coalescence-based species tree method. First of all, we wanted to be statistically consistent on the multi-species coalescence. We wanted to have these good theoretical properties. Then we wanted to be useful in practice. We wanted to be accurate. We wanted to actually give trees that are close to the true species tree. We wanted to be able to run on larger data sets. Now we have data sets, for example, the new 1KP data set, which I'll show some running time results on later, has over 1,000 taxa and hundreds of genes. And we also wanted to be fast on small data sets and, of course, easy to use. So we've designed ASTRID with these goals in mind. It's actually a very simple method. We start with gene trees, and we build these internode distance matrices. And all internode distance matrices, a particular element in an internode distance matrix corresponds to the number of edges between two taxa. So for example, if the internode distance matrix corresponding to this tree, there would be 1, 2, 3, 4 edges in the entry corresponding to A versus, in the A-B entry. Then we take all these internode distance matrices and we average them, just a regular average. And then we run a distance method on these. So the distance method that you're probably most familiar with is neighbor joining, but we use one of two methods, FastME and Bioinj star. And Bioinj star can run on distance matrices that are missing elements, which is nice for a lot of data sets. And then we record this estimated species tree. So this idea was originally used by NJS in 2011, which used neighbor joining to estimate the species tree. So the advantages of ASTRID over NJS is that ASTRID is a little more accurate because of the new distance methods like Bioinj and FastME, and it's much, much faster, as we'll see later. So it does have that nice mathematical property of statistical consistency under the multi-species coalescence. And what this means is that as we increase the amount of data we have, assuming we have true gene trees, which of course we don't really have true gene trees, but if we did, we could increase the amount of data, and eventually our error would drop to 0. We'd get a true tree with a probability approaching 1. And there's this nice proof of this in a recent paper. Of course, this doesn't mean that in practice it'll be accurate, and it doesn't say anything about the amount of data we need to get accurate trees. So we need to do experiments. And we use simulation studies to help us assess the accuracy of our method. So we start with a species tree. Sometimes it's a random species tree, sometimes it's a biological species tree. And we generate gene trees under a model of gene tree discordance. So in this case, we'll use ILS, but it's also possible that we could simulate things like lateral gene transfer. And then we simulate alignments using our model of sequence evolution. Then what we do is we plug this back into our gene tree estimation, or species tree estimation workflow. We estimate gene trees using a maximum likelihood estimator, in this case FastTree. And then we take that and we estimate our species tree with ASTRID, and we'll compare it to some other gene tree methods, or species tree methods, and then we'll compare it to the known true species tree. So this gives us a quantitative idea of the amount of error in our method. Just to recap, a lot of you probably know this already, but we compare trees by counting the number of branches that differ between the trees. So both these trees share this AB versus CDE split, but this tree has an ABC versus DE split, and this tree has an ABD versus CE split. So we'll call this 50% or 0.5 Robinson-Gould topological error, and that's the metric we'll show in a second. So we simulated this on a lot of data sets. I'm actually only going to show one of these data sets today because of the slightly shorter talk. But the point is we've tested on a wide range of number of sites, we've tested on a wide variety of number of genes, and we've tested on a wide variety of ILS conditions measured as the distance between the true gene trees and the true species tree. And that's gone all the way from about 10% to about 70%. So 70% is a quite high level of ILS, and we still get reasonably good performance under those conditions. So the first thing I want to show is a comparison to MPS. And MPS is a really popular species tree estimation method using the coalescent. And what I want to focus on is that both ASTRID and ASTRID, which are the two most accurate methods that we have for species tree estimation, are much more accurate than MPS, which is still widely used. Again, across a wide range of model conditions. You can see under this, this is a simulated data set based on the avian phylogeny that came out a couple years ago. With quite high ILS levels, nearly 50% of the branches in the true species tree are not in the true gene trees. And you can see that MPS has topological error, over 20% in some cases, whereas both ASTRID and ASTRID, have about half that. So we wanted to do a closer comparison of just ASTRID and ASTRID. Because again, ASTRID is a quite widely used, very accurate, and also quite fast method. So we did this on a whole lot of data sets and a whole lot of model conditions. But I'll just show you this one data set that's based on the mammalian simulated data Song et al. And what you see here is that ASTRID and ASTRID get nearly identical accuracy, topological accuracy, all the way from 10 genes to 200 genes. When we have these 500 base pair sequences. And we see this kind of across the board. And there are some cases where ASTRID gets slightly better accuracy, and there are some cases where ASTRID gets slightly better accuracy. But on the whole, their accuracies are quite competitive, and also quite, they're quite accurate. Once we get to 200 genes, we only have about 5% topological error, which corresponds to a small number of branches, 1 or 2 in this 37 taxon data set. We can extend this to the case where trees are missing taxa. So in a lot of cases, due to things like gene loss, we'll have trees on only subsets of the taxon set. So what we've done here is we've basically randomly removed taxa from our gene trees. So the whole taxon set has 50 taxa, but here we've removed 10, 20, 30, and 40 taxa from each tree. Here you'll notice we're using ASTRID Bioinformatics instead of ASTRID Taxa. This is because sometimes the distance matrix is missing elements, so we need a distance method that's capable of handling that, and there's a package called PhyDstar, which provides an implementation of BioNJ that can do that. So you can see that, first of all, ASTRID and ASTRID are getting very similar results. Second of all, even when we're missing a lot of taxa per tree, so we have 50 taxa, but each gene tree only has 10 taxa, right? And once we have enough genes, the error goes down quite a bit. It's down to 20%. When we have 1,000 genes, which is pretty good. Of course, not as good as when we have 40 genes per tree, but still pretty good. But the real benefit of ASTRID is it's accurate, but it's also extremely, extremely fast. So these are some running times on that 48 taxon avian data set, and we're increasing the number of genes from about 20 to 800. And you can see the NJS, which is the predecessor to ASTRID and ASTRID, both on this 800 data set, 800 gene data set, take about 5 minutes to run, a little less maybe. But ASTRID, and this is an older version of ASTRID, and I'll present some results with a newer version in a second, takes only a few seconds to run. So it's much, much faster, and it's capable of scaling to much larger data sets. So, for example, on a 1,000 gene, 1,000 taxon data set, that ASTRID takes 12 hours to run. Something like MPS wouldn't be able to run on it at all. And ASTRID with FastME takes only 0.008 seconds, or easier way to think about it is 30 seconds. Similarly, on the 1KP data set with 1178 taxa and 478 genes, ASTRID also takes about 30 seconds. And these are some of the largest data sets we have. So the fact that we're able to run these basically in, very quickly is promising. And if we think about the theoretical running time of ASTRID, it's also quite nice. The running time scales linearly with the number of genes, so if we have twice as many genes, it'll take twice as long, and quadratically with the number of taxa. So if we have twice as many taxa, it'll take 4 times as long. And this is, at least as good as most other species tree estimation methods. Most take longer than that. The especially nice thing is that the memory usage is constant with the number of genes, so you can add more and more and more genes, but you'll still be able to run it on your computer even if it'll take longer. You don't have to worry about running out of memory as you increase the genes. Of course, that's not true for taxa, but it's true for genes. So we can get, of course, we can run ASTRID on biological data sets. So, all right, there's no way we're going to be able to read this text, but we ran it on the mammalian data set with 37 taxa, and we get high brand support on almost all the edges. The only edge we don't get high support on is this placement of scant dead show, which those of you who study these organisms know is pretty contentious, whether it's closer to primates or clear. So this is a low support edge, and it's contested in the literature as well. But it is the same result that we get from ASTRID. It's the same result that we get from concatenated vaccine likelihood. And the really cool thing is we can run 100 bootstrap replicates on this data set with, there are 424 genes and 37 taxa in just 6 seconds. Whereas something like ASTRID, which is still quite fast, would take about 10 minutes to do the same thing. It's available in source and binary form. It runs on Linux, Mac, and Windows. Probably it'll run on your computer. Let me know if it doesn't. It's great when you have heterogeneity into ILS. So it's good for larger data sets. It's also good on smaller data sets, but it's actually designed for larger data sets. So I'd like to acknowledge funding and some references. And again, use ASTRID if you're estimating species trees. It won't take you very long. Thank you.